So this webinar will be on the selection of yeast and fermentation conditions for the production of Sauvignon Blanc. So as an agenda, I will make a short introduction on Sauvignon Blanc and then explain a little bit the impact of yeast on Sauvignon Blanc aromatic profile and some specificities. Then I will leave the speech to Marie-Charlotte COLOSIO for the selection and the screening of specific yeast for Sauvignon Blanc and the specific screenings. And then I will go back to you with a particularly interesting study case, that is the influence of yeast strains and fermentation parameters on Sauvignon Blanc profile. And then at the end, because we selected the yeast for that, I will show a little bit of the validation of this yeast and make a conclusion. So as an introduction, Sauvignon Blanc is one of the most widespread aromatic varieties throughout the world. It's actually the 11th most planted variety. And you all know that it's a South Hemisphere emblematic wines, especially in Australia, South Africa, and New Zealand. Sauvignon Blanc is actually characterized by very strong flavors, strong varietal flavors at the beginning. These strong varietal flavors are mainly two categories. The first category are the pyrazines. These pyrazines are really related to the date of harvest and let's say to the grape harvest. And it's the green, really green asparagus capsicum flavors. Then you have the other category of varietal flavors. It's what we call the polyfunctional thiols. The main things, actually, the main compounds are exposed here, 4-MMP and 3-MH, that impart actually box tree, blackcurrant, or grapefruit, passion fruit aromas. In the same times, you have strong varietal flavors, but it's balanced with fermentative flavors. So flavors coming not from the variety, but directly from the fermentation by the yeast. In that case, you have some higher alcohols with a strong impact, like 2-phenylethanol, the rose flavors. You have linear chain ethyl esters. So from butanoate to decanoate is the overall fruitiness of the Sauvignon Blanc and of the wines. And then you have very strong esters, so very strong aromatic compounds, acetate esters, and mainly isoamyl acetate with the banana and candy flavors. One of the compounds that is a bit different than the others actually is that the compromise between varietal flavors and fermentative flavors, and this is what we call the 3-MHA, so it's the 3-macaptohexyl acetate. This esterification is coming from the fermentation, but the substrate is coming from the 3-MH, that is a functional thiol. So actually on the Sauvignon Blanc, you have a strong yeast impact. The yeast impact and the fermentation conditions will impact all these compounds, so the polyfunctional thiols and the fermentative flavors. And we will see in the rest of this presentation this kind of impact. So to go on this impact on the Sauvignon Blanc, for sure, why we use a yeast? We use a yeast first to make the fermentation, so to ferment glucose and fructose into ethanol. But through this, let's say, glycolysis pathway, you can also have derived the fatty acid pathway and the fatty acid production. And we will see that it's very important for flavor production as well. The yeast, in the same time, during fermentation, is eating nitrogen source. Amino acids, ammonium, are actually one of the main nitrogen compounds that the yeast can use, and the yeast will use it, and in some pathway, like the Erdős pathway, will produce higher alcohols that are actually very aromatic compounds. From these higher alcohols and the acetyl-CoA that is produced through the fermentation pathway, you have an esterification that make the acetate esters. And these acetate esters have very strong flavors, as we see. So for example, if you start to take some amino acids as substrate, phenylalanine or leucine, and then they are going to produce the isoamyl alcohol or 2-phenylethanol as a higher alcohol. And these higher alcohols can be esterified with the acetic acid and go to produce 2-phenylethyl acetate and isoamyl acetate that are strong banana flavors and 2-phenylethyl acetate rose flavors. Among the fermentative aromas, you also have what we call the ethyl esters. These ethyl esters are actually coming from the fatty acid pathway, and this is the esterification between fatty acids and ethanol to make ethyl esters. And this is the overall fruitiness. So all these compounds are making fermentative aromas. On the Sauvignon Blanc, as we saw, you have also strong varietal flavors. But these varietal flavors can be either expressed or non-expressed. These varietal aromas are mainly the thiols, as we saw, but these thiols are in the form of a precursor in the grapes, glutathionylated or cysteiniolated, and they have to be uptake by the yeast and then converted into their volatile thiols, and these volatile thiols have the strong impact. One of these compounds is also coming, as we saw, from the fermentation and also from the substrate of the thiols is the acetate of 3MH with the passion fruit flavors. So if we focus a little bit more on the higher alcohols and the esters, this is here to come back. It's a bit complicated, but not that far. It's to say that are the main compounds produced during fermentation and responsible for fruity and floral aromas. But as you will see, these compounds are actually very highly regulated with a lot of genes involved either in their uptake for the permease, coding for the permease that you have from the nitrogen source, or the ATF1, ATF2 that are actually the acetyltransferase to produce acetate esters, or these genes that are actually fatty acid, ethanol transferase. And in that case, it's making ethyl esters. So it's very highly regulated, and depending on the yeast strain and the conditions, you will have either high production of higher alcohols and esters or low production of esters. As a sum up, for example, in this table, you have the main compounds here, 2-phenylethanol as a higher alcohol, acetate esters or ethyl esters, and you have their main catalyst and the expected influence of some of the fermentation parameters, temperature, initial nitrogen, and fatty acids. So for sure, these are expected influence. For example, for the isoamyl acetate, the temperature increase the evaporation, as we saw in the last presentation, and that's why it's quite negative for this compound. But for others, could be different. Anyway, all these expected influence have a high variabilities among strains, and that's what we will see afterwards. If we focus on thiols, so this is actually something bigger, a zoom on the thiol metabolism. So you have the uptake of the thiols and then the release of the volatile thiols from the precursors. These are the main compounds responsible for tropical citrus fruits and greenness. But again, all these compounds are highly regulated and depending on strains and conditions. And one of the main genes involved are, for example, IRC7 or STR3 for the sulfurlyase, so meaning what the enzymes that will cleave the precursor into the volatile thiols, and NTF1 to make the acetate esters. And one of the main regulation systems as well involved in the release of these compounds is actually the nitrogen catabolite repression, because this nitrogen catabolite repression will act on permease and will act on specific genes to express or don't express or lower express some of their activities. So regarding these thiols, you have about the same things. You have the same things here. So the precursors and the expected influence. We saw that, for example, the 3-mercaptohexyl acetate, though the 3-MH acetate is an acetate ester, have a high volatility, and this high volatility, when you actually ferment at higher temperature, you have more evaporation, and if you have more evaporation, you lose these kind of compounds. But again, these expected influences have a very high viabilities among strains, and this is what we will see in the following of the presentation. So some specificities. I said you have two specificities. The first specificity for thiol release is actually the IRC7 gene. If you see at genetic level, IRC7 gene is actually, you have one copy of the gene on both alleles, and this gene has a particular length. And if you have the full length, so IRC7 long, then it's transcripted into a long RNA, and this long RNA is then translated into a protein, and this protein is the IRC7P long with coding for an enzyme. It's an enzyme, so it's a carbon sulfur liase, and if the length of the IRC7 is okay, the carbon sulfur liase is active. So meaning that if you have the precursors like the cysteinated precursor of 4-MMP that come into this enzyme, then it has the possibility to be cleaved. And so the 4-MMP, the volatile thiols, can be released. The problem is that in some of the yeast strain, you have another version of the IRC7. It's the IRC7 short, so it's 38 base pair left. And in that case, this gene will code for an RNA that is a short, and it will be translated actually into another protein. And this other protein will have no carbon sulfur liase activity, will not be able to take the precursors and then to cleave the precursors, so will not be able to release the volatile flavors and so the corresponding aromas. One of the problems is that most of the wine yeast have a genotype that is short IRC7, short IRC7. So two copies of the gene IRC7, but a short one. So without the possibility to have a sulfur liase that is active. So it's quite interesting to see that there are different categories. You have strains with a genotype with full length IRC7 and some with only one allele with the long one, and others with only the short one, and that are most of the wine yeast. What we know as well is that the IRC7 is down-regulated by low temperature, and as well the activity of the enzyme. So that's the first specificity. The second specificity is, as we said, the nitrogen catabolite repression regulation system. As you know, the available nitrogen in the must is about 1/3 ammonium and 2/3 amino acids, the organic YAN, yeast available nitrogen. The yeast will use this nitrogen to produce biomass, to regulate the fermentation speed, regulate as well the completion of the fermentation, and then the flavor production and other metabolites. But there is systems that makes the yeast prefer the nitrogen source, the ones that are allowing its best growth. And in that case, it's actually what we call the nitrogen catabolite repression system. This system, as well as the SPS regulatory mechanism, they will actually be applied and regulate the uptake of nitrogen by the yeast to produce its best growth. So if we go a little bit more into details at genetic level, you have sequential consumption of nitrogen sources. First, you have the amino acids that are transported by specific permease and controlled by the SPS system. And then you have actually the ammonium and other amino acids, general amino acids, that are actually uptake by the yeast through permease, but these permease are downregulated by the nitrogen catabolite repression system. Meaning that as far as you have preferential nitrogen source, this will affect the uptake of the ammonium and other amino acids by the yeast. But ammonium is actually a bit specific. Ammonium is a bit specific because it's mediated by 3 permeases under nitrogen catabolite repression system. But we know as well that if you have a lot of ammonium in the extracellular medium, actually, as far as you have a lot of ammonium and a low level of other nitrogen compounds, it will be early assimilated. So it could prevent a little bit the uptake of other amino acids. So approximately you have the same order of consumption, but depending on the strains, it could be different. So the impact on thiols is very interesting. Why? Because of the uptake of the thiols, so the precursors of thiols are going through permease, and these permease are actually downregulated by or controlled by the nitrogen catabolite repression system. So as far as you have ammonium in a big source, it will delay a lot the uptake, or even block sometimes the uptake of the precursors of thiols, and it could be detrimental. Then you have also an action of the nitrogen catabolite repression on the bioconversion, because as far as this regulation system is active, it downregulates the IRC7 expression, and so don't express the activity of the carbon sulfur liase. So high nitrogen, active nitrogen catabolite repression, and so a possible decrease of first thiols uptake, but also bioconversion.